I'm Adam Algar from the University of Nottingham, and I'm here to talk to you about a paper that my co-author, LopezDarias, and I have coming out in the August 2016 issue of Ecography called Sex-Specific Responses of Phenotypic Diversity to Environmental Variation. Now, in this paper, we were interested in how different environmental conditions affect the ecomorphological diversity or the trait diversity of populations within a species. And we're interested in looking within species because differences, local adaptation between populations may represent the first stages in ecological radiation and maybe even in species diversification. And so, in particular, we were interested in how populations fill morphological space. So, bear with me. We can think of this balloon as the morphological space or the morphospace occupied by a species. So on one axis we may have body size, on another one head size or leg length. And environment may affect this morphospace in a couple of ways. First of all, in different environments, different trait combinations may be favored, and in which case we would expect to get a shift in the morphological space or the morphospace, in the position in morphospace that a population occurs. The second way that environment may affect this morphospace is that if, for example, a harsh environment imposes a filter on a population so that only a small subset of trait combinations can allow individuals to persist in that environment, in that case we would expect the filter to reduce the volume of morphospace occupied by that population, so it would have a much smaller volume in that environment. Now, to test these ideas, we needed a widespread species that's found across a diverse range of environments, and so we headed to the island of Tenerife. It's part of the Canaries Islands archipelago, and we worked on this kind of widespread lizard called Galodia galoti. Hopefully you can see it there. It's a lacertid, and on Tenerife, which is a very environmentally diverse island, it's found from kind of low elevation, hot, dry environments to slightly cooler, kind of wetter, low elevation environments, up to kind of 3,000 meters on the Teide volcano, which is a very extreme environment. It gets snow in the winter, and in the summer it's very dry with very high solar radiation and can get very hot. Now, traditionally in ecological studies that have looked at how ecomorphological traits vary between environments, we treat populations as single entities, but actually within populations, a very big difference between individuals is which sex they are, and often males and females can be very different. And so one of the particular things we wanted to do in this paper was actually test whether males and females respond similarly to environmental variation. So bear with me. We looked at males and females separately. So here's our males. And here's our females. And so within a population, we have the morphospace occupied by males and that occupied by females. And we looked at how populations, males and females, shifted in morphospace between low elevation and high elevation environments, and also how the volume of morphospace occupied was affected. And we found a couple of really interesting things. One being that as we move from low elevations to high elevations in males, we found shifts in morphospace. And we also found changes in the volume occupied. And so it looks like for males, there's a strong filter operating at high elevations, where males tend to be much smaller than they are at low elevations with relatively larger heads. This was in real contrast to females, which regardless of where they occurred on the island and which environment, they were broadly occupied the same morphospace. They didn't move around much and their volume didn't change. So this had a couple of side effects, one major one being that at high elevations we had much lower sexual size dimorphism as males and females became more similar. So what does this tell us? Well, first of all, I think it tells us that it might be when we look at how populations diversify across very different environments, natural selection may not be the only part of the story. These differences between males and females seem to implicate something to do with sexual selection gradients affecting males, provided the changes we saw are adaptive. And more generally, I think it demonstrates that in macroecological or ecological studies, when we're looking at things like functional diversity, If we treat males and females as identical, then we may be missing an important source of variation that may actually, in some cases, enhance or sometimes constrain functional diversity and diversification. So thanks for listening. Check the paper out in the August 2016 issue of Ecography, and thanks a lot.